Hello, IB Bio students. In today's lesson, you are going to be looking more at the ultrastructure of cells, just as we've been doing in class. And in that process, you're also going to be comparing prokaryotic and eukaryotic cells. In today's activity, instead of looking at cells under a light microscope, you are going to look at electron micrographs, which are pictures that were created using an electron microscope. If you remember from your last virtual lab, electron microscopes are those that use electron beams focused by electromagnets to generate images of specimens. They have two major advantages over light microscopes in that they can produce higher magnification, so we can see smaller things, and they enhance or increase resolution of pictures, so the pictures look clearer as you see them, so the structures within the cells will look clearer to you on an electron micrograph. The major disadvantage is that all specimens have to be dead for an electron microscope to work, so you cannot look at living things with an electron microscope. With our light microscopes, we can easily see cells and some of the structures within the cells. With an electron microscope, we will be able to see even smaller structures within cells. You can also see such small structures as viruses, molecules like DNA or sugars, and at some point you can even see down to the atomic level. Your primary goal for today is going to be to complete this activity titled 1.2 Electron Micrograph Lab 1. And some of these activities here will ask you to practice some skills you've already done, and some will ask you to learn or practice new skills. Question number 1 says, using the scale given in a ruler, A, calculate the magnification of the image, and B, determine the actual diameter of the nucleus in micrometers, drawing a line to show my measurement, and showing work all through. So just thinking about this to begin with, I'm looking at this electron micrograph, and it's a portion of a cell, and I can tell that this structure here in the middle is the nucleus. So I know I'm going to need that at some point in time. Way down here, almost hidden in the corner, is a scale bar, so I know I'm going to need to use the scale bar at a certain point in time also. So those are some hints to get you started. The other thing that I want to make sure that you see is this instruction here, draw a line to show your measurement. So when you get ready to do the nucleus portion of this, make sure you take a ruler, lay it across the nucleus, and draw your line. And so it's very obvious to me where you made your measurement, and that will be what you will need to measure when you're working at the diameter of the nucleus for part B. Question 2 asks you to do some labeling, and it says it's a prokaryotic cell, so this is a bacteria. Notice that it gives you the terms that you're to use for labeling this. And then it says draw and label the cell in the box. So you're probably wondering to yourself, why in the world do I have to draw and label a cell that's already pictured for me? Well, the reason you have to do this is that one of your assessment statements asks you to be able to do it, and so this is your practice in drawing one of these cells. So at this point, you'll need a reference to help you with this, and you have your note sheets that you worked on in the last class period that you can use, or you can always use your textbook, your online textbook, the course companion. So I'm going to go there, and I'm not quite sure how to access it, so my first stop would be our Canvas page. And as I scroll through the Canvas page, I'm not seeing any obvious links, so I'm going to click right here where it says Important Information for IB Bio. And here I see Course Companion eText Login page. So I'm clicking on this, and I have to click a second time to get to my login page. And so you should have already set this up so you can access the Course Companion. And you'll log in, and this looks like our Course Companion, so I click on it. And here's my book. Well, I don't want to have to flip through page after page, so I'm going to click down here at the table of contents. I know we're on section 1.2. So here I am at section 1.2. I don't see a whole lot on here that will help me label that prokaryotic cell, so I'll go to the next page, and here I see a nice section on drawing prokaryotic cells. So I see some help with the actual drawing, I see some help with the labels, so this will be very helpful to me in labeling and drawing that prokaryotic cell. Last thing about this, I know on the outside of a prokaryotic cell there are often 3 layers. The capsule, the cell wall, and the plasma membrane. So when I draw my cell over here, I want to make sure that I show 3 distinct layers on the outside, and I make sure that my label lines actually touch those 3 distinct layers. Progressing through this activity, Numbers 3 and 4 look at electron micrographs from first a plant cell and then an animal cell. So I'm looking at this plant cell here, and I know I need to label these parts. Up here again, I have what my answer choices are, so be careful, make sure you use the actual answer choices. Question 4 is asking the same thing, only over an animal cell, and again, the answer choices are given. So if I go back to my course companion, this box is on prokaryotic cells. That won't help me a whole lot with plant and animal cells, as those are both eukaryotic cells. So I'm going to go forward one more page, and here I see a section titled Drawing Eukaryotic Cells. Well, I'm not drawing at this point, but I'm labeling, but these pages give me really nice pictures to show me what I'm looking for. So I can see here that this is what a nucleus looks like, this is what rough ER looks like, and if I'm not happy with the size, I can go down here and I can zoom in, and so I can see the picture's a little larger, and I can keep doing that if I would like. If I go to the next page, here are more structures that I would find in typical eukaryotic cells and what they actually look like in an electron micrograph. And over here are acceptable ways to draw them and some descriptions of the things. So this will be very helpful resources for you as you're working through numbers 3 and 4 in the electron micrograph activity. The last page of this activity is a little bit different, and this is where students often don't read the directions very carefully and don't complete all the activities. So in each box there is a portion of an electron micrograph, and it says identify the cell structures pictured, then illustrate one of the cell structures shown in each picture and label it. So again, one of the assessment statements has to do with being able to draw from electron micrographs, so we're practicing this. So as I look at this very first one, I can see the large structure here is a mitochondrion. I can see some ER around it, and there's little dots on that ER, so I'm assuming those are ribosomes, and that's rough ER. Right down here, I may even see the beginnings of Golgi body, and of course everything around those organelles is cytoplasm. So the cell structures that I see that I'm going to write in my box, I see mitochondria, I see Golgi, I see rough ER, and I see cytoplasm for sure. So I've got all those things. Not quite sure why it's jumping around on the screen for me. But then I'm going to pick one thing to draw, and I'm going to go ahead and draw mitochondrion. And I know mitochondria is two membranes, so I've just drawn the outer membrane. The inner membrane is continuous, and it's what creates the little structures that you see going into the inner portion of the mitochondrion. So this is actually the inner membrane of the mitochondrion. And then I should label that this is the mitochondrion. So that's how you will go about completing this activity. If you get finished, you should call up the second electron micrograph activity and work on that. That one we will also take a look at in class. Make sure that when you're looking at that one that you look at the color version on the screen as well as the black and white version that you have in front of you on paper.